B. Prevention, diagnosis and cure of disease. Prevention of many genetic disease in early period of pregnancy will help the mothers to give birth to the healthy babies. Moreover, certain viral, e.g. polio, AIDS, acquired immune deficiency syndrome, and protozoan diseases can be prevented from infection in humans. 1. Prevention of disease. For preventive measures, several immunogenic polypeptides, vaccines, and proteins, antibodies, have been chemically and biologically synthesized. Moreover, it is expected that in near future immunogenic compounds would be available in market and more information would be on cloned genes for viral, AIDS, bacterial cholera, and protozoan malaria diseases. 2. Diagnosis of disease. Secondly, genetic engineering techniques have solved the problem of conventional methods for diagnosis of many diseases. DNA probe, monoclonal antibodies, and antenatal diagnosis, some of the available methods used as a tool to diagnose a particular disease. A. Diagnosis of parasitic disease through DNA probe. Probes used to diagnose pathogens contain the most specific DNA sequences of genetic material of parasite. The lies in such a way that the other related species or strain do not contain those sequences. These unrelated unspecific sequences of parasite are first recognized by using DNA hybridization technique. Then a DNA sequence, not present in any species, is identified, cleaved by using restriction enzymes, and inserted into a cloning vector plasmid. The bacterial cells are transformed by the recombinant vector. The transformants are multiplied. Finally, the foreign DNA fragments are retrieved from the host cells. The DNA sequences of the parasite, thus obtained, are labeled with radioisotope and used as a probe. The probe can also be chemically synthesized. Following are the steps for diagnosis of a particular disease. 1. Isolate the parasite from the infected tissue of the patient. Extract the DNA from parasite and purify it. 2. Break the DNA by using restriction enzymes to get the DNA fragments of varying size. 3. Electrophorese the DNA solution of different length by using agarose gel just to get a smear of DNA. 4. Attach the DNA smear to more firmer support by southern blotting techniques. Thus the filter paper carries the exact replica of the DNA adhered to it. 5. Hybridize the immobilized DNA on filter paper by incubating it with radiolabeled probe. Probe DNA complementary to certain DNA sequences of parasite DNA sticks to it and forms the hybrid, and 6. Wash the filter paper to remove unhybridized probe. Keep the filter in contact of X-ray film. Dark bands appear where DNA probe had hybridized the specific target sequence. Thus, a parasitic disease is diagnosed positive. If dark bands on X-ray film do not appear, the absence of parasite is noted. Specific DNA probes have been designed which have shown good results in diagnosing the infection caused by viruses, bacteria or protozoa. Tuberculosis (TB), caused by Mycobacterium tuberculosis, is one of such diseases which can be diagnosed by this method. Moreover, a complete testing system for TB is marketed by GenProbe, Inc., California. Similar efforts have also been made for diagnosis of leprosy, Kala Azar, caused by Leishmania donovani, malaria, etc. B. Monoclonal antibodies. Antibodies are proteins synthesized in blood against specific antigens just to combat and give immunity in blood. They can be collected from the blood serum of an animal. Such antibodies are heterogeneous and contain a mixture of antibodies, i.e. polyclonal antibodies. Therefore, they do not have characteristics of specificity if a specific lymphocyte, after isolation and culture in vitro, becomes capable of producing a single type of antibody which bears specificity against specific antigen. It is known as monoclonal antibody. Due to the presence of desired immunity, monoclonal antibodies are used in the diagnosis of diseases. Major difficulties with antibody-secreting cells are that they cannot be maintained in culture. But myeloma cells, bone marrow tumor cells due to cancer, grow indefinitely to produce a huge quantity of identical cells clones, and also produce immunoglobulins in the same amount. The immunoglobulins are in fact monoclonal antibodies. 1. Hybridoma. Cesar Milstein of Argentina tried to culture myeloma cells. In 1973, in collaboration with Cotton, he succeeded to fuse rat and mouse myeloma cells with the result of production of hybrid cells. The hybrid cells secreted the immunoglobulins which consisted of several types of polypeptides. In 1974, George Kohler and Milstein successfully isolated clones of cells from the fusion of two parental cell lines, i.e., between lymphocyte from spleen of mice immunized with red blood cells from sheep and myeloma cells. These cells were maintained in vitro and produced antibodies. The antibodies for specific antigens immunized the myeloma cells. Moreover, the hybrid cells maintained the character of lymphocyte to secrete antibodies, and of myeloma cells to multiply in culture. These hybrid cell lines, cell clones, are known as hybridemas, which are capable of producing unlimited supply of antibodies. For the discovery of production of monoclonal antibodies, Kohler and Milstein along with Niels Jerne were awarded Nobel Prize in 1984 in physiology and medicine. Now, techniques are developed to obtain antibodies from hybridoma culture, figure 7.6. Monoclonal antibodies are produced against a variety of proteins, glycoproteins, glycolipids, nucleic acids, and chemically defined groups linked to protein carriers. Possibility O. Success is related to the number of antibody-forming cells in spleen at the time of fusion. This, in turn, depends on immunogenicity of the antigen used. For example, a mouse spleen contains about 10 to the power 8 cells. Sheep red blood cells, SRBC, act as immunogens and stimulate the replication and differentiation of our body-forming cells. At the time of secondary response, each spleen may contain 10 to the power 6 cells producing antibody against SRBC. Among 2x10 to the power 5 spleen cells, only one hybrid cell is generated. About 500 hybrid cells per spleen can be obtained, where 5 of these produce specific antibody. The cells growing on culture and producing sufficient amount of monoclonal antibodies can be frozen. The cells are collected through centrifugation and resuspended at a density of 10 to the power 7 cells per milliliter freezing medium in cold at 0 degree. Samples 0.7-1.0 milliliters poured in sterile glass vials are sealed and stored at 80 degree. See figure 7.6. Outline of production of monoclonal antibodies. In India, a hybridoma laboratory has been set up with indigenous material at the Research Institute, Creek, Bombay. The first experiment conducted at CRI was an attempt to develop leukemia cell-reactive monoclonal antibody. Uses. Monoclonal antibodies are useful in the following cases. 1. In diagnosis of ABO blood groups, cancer, pregnancy, allergies, viral diseases, and certain hormones. See antenatal diagnosis of HAEMOGLOBINOPATHIES. There are as many as 2,500 genetic defects known to occur in human beings. Some of them, e.g., phenylketonuria and hemophilia, have been cured, but most of them are not understood. If a pregnant woman bears a child with a genetic defect, often she is advised to abort it but not to give birth to genetically defected child. Thus, the technique of diagnosis and thereby suggestion for abortion of a genetically defected child is known as antenatal diagnosis. The genetically determined defects of function or synthesis of human hemoglobin is known as hemoglobinopathies. The inherited hemoglobinopathies are 1. The structural variants, where till now 300 amino acid variants are identified, and 2. The thalassemias, a condition characterized by reduced or absent synthesis of a cis globin chain, A and cis thalassemia respectively. A. Methods of antenatal diagnosis. Antenatal diagnosis is done by taking a few milliliters of amniotic fluid from the fetus of about 16 to 18-week pregnant women. Fetal blood samples are analyzed by testing for the ability to synthesize globin chains, alpha and beta. Difficulties with the antenatal diagnosis are that the fetus of less than 3 months of age synthesize very little alpha or ceobin chains. At this stage, the fetal blood contains normal level of and low level of sus chain. Synthesis of sus chain takes place in low amount in fetus of 8 weeks age, and therefore, it is possible to analyze the euglobin in a fetus but difficult to analyze sus chains. The fetus fluid also contains photoly-derived fibroblasts which serve, with or without additional culture, as a source of tissue for preparation of fetal DNA. Structural analysis of DNA is done by 1. Isolation of all the globin genes as recombinant DNA molecules, and cloning in bacteria, or 2. Without cloning. The recombinant DNA in bacteria directly by Southern blotting technique, and 3. Linked polymorphism. There are about 35 diseases which have been identified by antenatal diagnosis. This method helps in suggesting the abortion of defected baby, and if possible, to cure. It can be recommended for gene therapy as well. Thus, the antenatal diagnosis is applied for both genetic counseling and gene therapy. B. Genetic counseling. Genetic counseling is a technique which is carried out in detail based on antenatal diagnosis and suggested for the possibilities of future progeny. A genetic disease is first identified by a genetic counselor who understands the family history of genetic disease, and then suggests for the possibilities of giving birth to the child or aborting the child. The child may be normal, genetically defected, or carrier of genetic disease. Take the case of thalassemia. Alpha thalassemia is caused by the presence of two chromosomes. Each of them carries one dysfunctional globin gene of one normal chromosome and other dysfunctional genes. Undoubtedly, a person carrying two chromosomes each with single functional genes donates one normal genes to their progenies. It is, therefore, impossible to suffer their progenies from hydrops fetalis as it occurs due to complete dysfunctioning of all genes. DNA analysis of thalassemia has revealed that non-function of both genes on a single chromosome is only due to deletion of both genes from the chromosome. Recent techniques have made it possible to detect a thalassemic couple for the presence of no gene chromosome. However, there is risk for giving birth to hydropic fetuses if both of the couple have a thalassemia due to no gene chromosome. If there lies any possibility for gene therapy, the patients are also suggested for the same. It has also been suggested that one should go to genetic counselor before marriage for safe and normal progeny. 3. Gene therapy. There are many diseases which can be cured by using specific medicines synthesized biochemically. Nowadays, techniques have been developed to produce recombinant therapeutic biochemicals, for example, insulin, interferon, somatotropin, somatostatin, endorphin, human blood clotting factor 8, C, immunogenic proteins, etc. After 1975, a remarkable advancement in recombinant DNA technology has occurred and accumulated such knowledge that has made possible to transfer genes for treatment of human diseases. Several protocols have been developed for the introduction and expression of genes in humans, but the clinical efficiency has to be demonstrated conclusively. Success of gene therapy depends on the development of better gene transfer vectors for sustained, long-term expression of foreign gene as well as a better understanding of gene physiology of human diseases. Genes are the ultimate molecular switches that control various cellular processes. The abnormal gene expression can manifest in the form of specific genetic disorders. Until the last decade, delivering genes into humans to correct diseases has been accepted as scientifically viable and recognized as an independent discipline and christened gene therapy. The ultimate goal of gene therapy is the gene replacement therapy. Gene replacement therapy permits physiological regulation of the transgenes and elimination of the possibility of insertional activation of other cellular genes which occur at the time of random integration of the foreign gene. At present, the current strategy for gene therapy largely centers around gene augmentation therapy, where the foreign gene replaces the defective or missing gene. Overall, there are two strategies for gene transfer. One, the in vivo approach, which involves introduction of genes directly into the target organs of an individual. It is done in patients, therefore, also called patient therapy. 2. Ex vivo approach, where cells are isolated for gen transfer in vitro followed by transplantation of genetically modified cells back into the patients. A. Types of gene therapy. All the gene therapies that can be done in humans and classified into the following 4 types. 1. Somatic gene therapy. The genetic defects are corrected in somatic cells of the. It was initially formulated for the treatment of monogenetic defects but now holds promises a wide range of disorders such as cancer, neurological disorders, heart diseases and infectious diseases. Table 7.2. Sufficient expertise in performing successful gene transfer in somatic cells is required before carrying out gene manipulation in humans. See the table 7.2. Genetic disorders and acquired diseases amenable to gene therapy. CFTR, cystic fibrosis transmembrane regulator, SCID, severe combined immunodeficiency syndrome, DMD, Duchenne muscular dystrophy, ADA, adenosine deaminase, HSV-TK, herpes simplex virus, thymidine virus, NGF, nerve growth factor TH, tyrosine hydroxylase, TPA, tissue plasminogen activator. 2. Germ-line gene therapy. The functional genes are introduced into the germ cells for correction of genetic defects in the offspring. This therapy is being carried out in laboratory and farm animals. However, it has not been attempted in humans due to technical and ethical problems. One of its types is the embryo therapy, where embryos are diagnosed for genetic defects. If any such disease is present, the patients are advised for embryo therapy or abortion. In young embryo, a functional gene is transferred through microinjection technique. 3. Enhancement genetic engineering. This type of gene transfer is done for the improvement of a specific trait in animals, for example, introduction of growth hormone gene to increase height. It is being carried out in laboratory and farm animals. 4. Eugenic genetic engineering. Novel genes can be introduced in humans to alter or improve complex traits such as intelligence and personality. This type of therapy is not being attempted in humans because it is far beyond our technical capabilities and ethical problems. In 1990, for the first time, Michel Blees and W. French and Resco of National Institute of Health, Bethesda, USA attempted gene therapy on a human patient. A 2-year-old girl was suffering from severe combined immunodeficiency, SCID. This disease is caused by a faulty gene which expresses the enzymes adenosine deaminase, ADA. Deficiency of ADA results in the production of a chemical which selectively destroys the TUN-sus cells of the immune systems. Finally, the patients die. The scientists introduced a healthy ADA gene into the body of the girl who protected her immune system from damage. This successful trial has given the signal for the dawn of a new era in the field of medical sciences. B. Methods of gene transfer. A variety of gene transfer strategies have been developed during the last decade for the treatment of human diseases, which can be grouped into the two major categories, the viral and non-viral methods. 1. Virus vectors. After 1980, much work has been done on retroviruses as gene transfer vectors, more specifically on murine leukemia virus (MLV) for gene therapy. Efforts are being made to develop HIV-based vectors so that even non-dividing cells can be injected. The outline of retroviral gene transfer has been shown in Figure 7.7. The steps of developing a replication-defective recombinant retroviral vectors are: I. The replacement of viral structural genes e.g. GAG, POL and ENV by the therapeutic foreign genes of interest. E. Transfection of this vector into packaging cell line, i.e. producer cells that provide the viral structural proteins in trans so that the recombinant retroviral genome is packed and replication-defective retroviruses are generated. E. Transfection of host cells by such viruses, and reverse transcription of recombinant retroviral RNA and random integration into the host genome. In the absence of viral genes, the foreign gene therapeutic in nature is transcribed from the viral LTRs, the long terminal repeats, and desired protein is synthesized. Figure 7.7. The retroviral vectors are used in ex vivo gene transfer experiment, although it has been shown that they can infect a regenerating liver when administered intravenously into hepatectomized animals. See Figure 7.7. Retroviral gene transfer. A construction of a retroviral vector. B. Production of recombinant retrovirus. C. Transfection of target cells. D. Synthesis of recombinant protein. Adeno-associated virus AAV is a non-pathogenic parvovirus of humans. It has aroused interest as a vector for gene therapy. It requires a helper virus co-infection for viral replication. The potential use of this virus as a gene transfer vector has intensified research on the life cycle of the virus. An outline of introduction of ADA gene into the bone marrow cells, lymphocytes, is given in Figure 7.8. ADA gene is fused with an antibiotic G418. G418 facilitates in the selection of ADA gene. Therefore, ADA G418 is cloned into a plasmid containing proviral DNA from a retrovirus. The retroviral genome is defective. Some of the proviral DNA is replaced with the ADA gene. The recombinant DNA is then introduced into fibroblast cell line. A fibroblast cell line harboring the missing gene is used to produce the mature viral particles upon transfection with the recombinant DNA. When the mature viral particles are released, bone marrow cells are added to growth medium. In turn, the viral particles infect bone marrow cells, lymphocytes. Bone marrow cells, lymphocytes are then selected by using G418 antibiotic in growth medium. Thereafter, the cells are transferred to a second flask containing G418 in growth medium. In this way, the selected bone marrow cell lymphocytes are then re-injected back into experimental mice or for trials in children. 2. Non-viral approaches. Most of approaches in gene therapy has been focused on development of viral vectors rather than clinical efficacy and biosafety. Furthermore, technique of virus-based gene therapy is very costly and complex. Therefore, this led to the development of non-viral gene therapy. Majority of non-viral methods follow the in vivo approaches. Therefore, a gene can be used successfully as a drug. In general, the non-viral methods for gene transfer can be grouped into two methods: physical and chemical methods. Physical methods. The various forms of physical methods are given below. Microinjection. It is a very tedious method. However, it is used in oocytes, eggs and embryos. In 1993, Jeffrey S. Chamberlain and co-workers from of Human Genome Center, Michigan University, USA have cured mice that inherited a neuromuscular disease which is similar to muscular dystrophy of humans. The mutant mice lacking 427 Kelvin's gene in brain and muscle cells had a weak diaphragm which gradually degenerates like that in dystrophy-affected humans. Chamberlain and co-workers microinjected the DNA containing 427 Kelvin's gene into the zygote of mutant mice. The transplanted genes worked properly and produced necessary protein and thus prevented the diaphragm. Direct DNA injection. Direct injection of DNA into skeletal muscle has created a lot of excitement and led to the possibility of using gene as vaccines. Direct gene transfer was excitement and led to the possibility of using gene as vaccines. Direct gene transfer was attempted since early 1960s, but these studies were not done seriously due to inefficient gene transfer and low level of expression. Due to low level of expression, therapeutic benefits for the treatment of genetic disorder could not be derived. Thereafter, these studies were further carried out. The low levels of foreign proteins synthesized in the host cells were processed along the class 1 MHC pathway, leading to presentation of peptides on cell surface, resulting in a potent immune response against the foreign antigens. This gave the birth to the concept of DNA vaccine or genetic immunization. Rangarajan and Padmanabhan, 1996. See earlier section. DNA vaccines. Receptor mediated gene delivery. It is an attractive strategy. It takes advantage of normal physiological pathway. The receptors are exclusively present on hepatocytes. They bind to certain glycoproteins lacking the terminal azelaic acid. The concept of delivering genes into specific tissue was examined extensively with the liver azeloglycoprotein receptors. An azeloglycoprotein, orosomacoid, is coupled with poly-L-lysine. This conjugate is then condensed with plasmid DNA via electrostatic. See figure 7.9. This soluble complex, i.e. azeloglycoprotein poly-L-lysine DNA complex, ASGPPL DNA, is injected intravenously, intraperitoneally, which gets entered into hepatocytes via the azeloglycoprotein receptors, ASGPR. DNA is delivered into the nucleus. The transgene expression occurs, therefore, recombination protein is detected for several weeks. In 1991, G.Y. Wu and co-workers later extended the other cell surface receptors such as transferrin receptor. The major problem related to receptor-mediated gene delivery system has been the degradation of DNA in the endosomes by lysosomal enzymes. Therefore, co-administration of lysosomotropic agents, e.g. chloroquine, is done to enhance gene expression. Moreover, it is a established fact that adenoviruses escape lysosomal degradation by acidification of endosomes. Methods have been developed to construct adenovirus-linked molecular conjugate vectors such as monoclonal antibodies against a heterologous epitope on the hexane protein of the adenovirus envelope, are covalently linked to polylysine. This complex is coupled with plasmid DNA. Delivery of such molecular conjugate has been shown to facilitate more efficient gene transfer than the conventional receptor-mediated gene delivery strategy. Such a conjugate interacts with adenoviruses that express the epitope on the envelope. Embryo therapy through IVF technology. In 1993, adopting IVF technology, a Handyside, a team leader of British researcher at Hammersmith Hospital, London, got success in producing a genetically engineered female baby, whose parents transmitted the genetic disease, cystic fibrosis, in earlier 4 babies, who died later on. Even after confirmation through prenatal diagnosis that the babies would be sufferer or carrier of this disease, they decided to give birth to babies, which failed. This disease clogs the lungs of sufferer and makes them unable to digest the food properly. Handyside admitted 7 couples. Their eggs were collected and fertilized with their respective husbands' sperm in test tubes. After 3 days incubation in vitro, when embryo reached 8-cell stage, a single cell was removed for analysis. The cell was magnified 1 million times by using a magnifying thermal cycler. The individual defective gene which causes cystic fibrosis was identified and screened out. Thereafter, the embryo was implanted in the womb of mother. Consequently, a female baby, C. O'Brien, was born who was neither sufferer nor carrier of this diseases, but a normal one. The birth of this genetically engineered baby has increased the hope of millions of couples who transmit the genetic disease to their offsprings, and also of researchers to look upon many more genetic diseases to cure. C. Success of gene therapy. The success of gene therapy depends on gene delivery mechanism as well as on the choice of target tissue. In 1996, P. N. Rangarajan and Padmanabhan have discussed different conditions leading to success of gene therapy. 1. Cell types capable of dividing in vitro, e.g., myeloblasts, hepatocytes, keratinocytes, endothelial cells, etc., are amenable for in vitro and in vivo gene therapy. Both the in vivo methods are preferred for cell types such as neuronal cells. 2. The function of gene products also govern the selection of tissues. For example, in case of hemophilia, a gene can be delivered in any tissue provided the gene product is released into bloodstream. In addition, in case of cystic fibrosis, the gene should be delivered to specific cell types where introduction of correct gene is required. 3. Another attractive strategy for the treatment of several disorders is antisense gene transfer. For example, in SAS thalassemia, globin chains are accumulated in RBCs that result in their premature decay. Such types of destruction can be prevented by infection of K562 erythroleukemia cells with AAV expressing human globin gene in antisense orientation. 4. The potential of a gene delivery system is first found out in cultured cells or laboratory animals by using reporter genes for luciferase, growth hormone, SAS galactosidase, etc. For detailed discussion on reporter genes, see Chapter 9. On the basis of result of these studies, again pretrials are conducted in laboratory animals to test the level and duration of expression of the introduced genes. Finally, clinical efficacy is evaluated in human patients. But before conducting clinical trials in humans, permission is necessary from the regulatory agencies such as Recombinant DNA Advisory Committee (RAC) and the Food and Drug Administration (FDA) in the USA, the Gene Therapy Advisory Committee (GTAC) in the UK, etc. In India also guidelines have been formulated. See Chapter 22, Biosafety. Clinical trials on gene therapy are given in Table 7.3. D. Future needs of gene therapy in India. Gene therapy has tremendous scope for treatment of several disorders such as cancer, cardiovascular and neurological disorders. To initiate gene therapy, the functional studies should be focused on 1. Design of newer vectors for gene delivery, newer approaches to systemic delivery, 2. Targeting to specific tissues and cells, 3. Stability and duration of expression of gene introduced, 4. The statutes of the introduced genes in vivo, 5. Design of appropriate animal models, 6. Assessment of risk-benefit states, and 7. An understanding of molecular basis of cellular humoral immune response in case of DNA vaccines. The need of ours is to start more studies in different systems on various aspects, realizing the significance of approaches of gene therapy. Department of Biotechnology, Gov of India has constituted a group of experts to activate this area of research. At present, there are only 3 to 4 laboratories in this country that have started work on this area. At South Campus of Delhi University, in 1994, Bajaj and Sarkar have demonstrated the delivery of macromolecules such as toxins, drugs and nucleic acids into viable cultured cells through the use of reconstituted Sendai virus envelope (RSVE). This approach needs to be extrapolated under in vivo conditions. At the Cancer Research Institute, Mumbai, treatment of oral cancer through gene targeting to oral mucosal cells has been demonstrated. In 1995, at the Indian Institute of Science, Bangalore, Prabhu and co-workers have demonstrated the reporter-mediated targeting and expression of growth hormone gene in rat liver by using a regulatable promoter, CYTOCHROMEP450 promoter induced by phenobarbital. In 1996, Rangarajan and co-workers have demonstrated a newer approach to delivering DNA to hepatocytes by perfusion of liver with DNA lipofectin complex. See DNA profiling fingerprinting.